For my cell, I did a plant cell. So the cell wall is composed of cellulose. Cellulose is a structural carbohydrate and is considered a complex sugar because it is used in both protection and structure. It determines what comes in and out of the cell. So for my cell wall, I decided to use frosting because it protects the cake from air, causing the cake to not dry out. For the plasmomatas, I decided to use the lifesavers because the plasmomatas are holes in the cell wall that allow for nutrients to enter the cell wall as waste to exit the cell wall, and lifesavers have holes in them, so they allow for things to enter and exit the cell. For the plasma membrane, it is a boundary between the cell and its environment. It regulates what enters and exits the cell. I used a darker colored frosting for this because the frosting creates a boundary from the air and allows the cake to stay moist. So the nucleus has two major functions. It stores the cell's heredity material and DNA, and it coordinates the cell's activity. So I decided to pick a plum because it resembles the same shape of the nucleus, and it also holds DNA of its own. As you can see, this right here is the nucleolus. The skin is the nuclear envelope, and the chromatin is the inside of the plum. The nuclear envelope surrounds the fluid portion of the nucleus, called the nucleoplasm. The ribosomes are on the outer surface of the envelope surface as sites for protein synthesis, along with the ribosomes located in the cytoplasm. In my cell, it will be the outer part of the plum skin, so right here as I showed you. The nucleolus is the inside right here. So it is a round body located in the nucleus of the cell, and it is not surrounded by a membrane, but it sits in the middle of the nucleus. It also makes ribosomal units, subunits, I should say, and RNA. It also makes protein in the cells. So in my cell, you can see that I used the seed of the plum. There's little grooves in it, like here and here, that represents the nucleolus pores that allows for the protein to come in and out. It has the similar shape, round shape, as a nucleolus. That's why I decided to pick it. So the chromatin is what packages DNA into a smaller volume that fits into the cell to control the gene expression as well as DNA replication, and to strengthen the DNA to promote mitosis and meiosis while preventing DNA damage. In my cell, it is all this inner sides of the plum. So rough ER and smooth ER. So rough ER is a membrane system that folded sacs interconnected channels that serve as a site for protection in lipid synthesis. This is a type of ER that is studied with ribosomes. So as you can see, here are some of the folds in the rough ER, and these little dots right here are the ribosomes. The smooth endoplasmic reticulum regulates and releases calcium and protects toxins. So this right here is smooth. It doesn't have any ribosomes like this one does. I decided to use airheads because I can easily form them to a similar shape as the both smooth ER and rough ER. Also because the smooth ER and rough ER are the same type of membrane makeup, I can use the same thing, but I can add ribosomes onto the rough ER. Ribosomes, they're structures, cellular structures that perform translation or photosynthesis. They read an mRNA sequence, translate it, and use tools to build it into a protein. They also are free in the cytoplasm, so I decided to use sprinkles. They're similar in structures, just like the ribosomes. The cytoskeleton, it forms a framework for the movement of organelles around the cytoplasm. Most of the organelles are attached to it. It consists of protein, microfilaments, and intermediate filaments, and microtubules. So I decided to use pull-apart Twizzlers for this because they're similar to tube-like structures, just like the cytoskeleton. For the Googlio apparatus, I used gummy worms. Unfortunately, there were mini ones. I decided to use it because it looks similar to the Googlio apparatus structure, and the Googlio apparatus structure is job. It's supposed to package and sort proteins to send them to their destinations. They're normally stacked up on each other, as shown in the picture. However, the stack amount can range. So the central vacuole, I used a lemon. It's only found in plant cells, and it is surrounded by a membrane, and it functions to hold materials and waste. It also functions to maintain the proper pressure within the plant cells to provide structures and support for the growing of the plant. Why I decided to use a lemon is because it helps clean the body of toxins. So for this, it would be the cells holding waste, and it also helps the body reduce bloating. So in this case, the cells maintaining the proper pressure so it does not burst. Chloroplast. So what I decided to use for chloroplast is green food coloring and some green sprinkles, and this is frosting because the green sprinkles and food coloring provide the frosting with a green pigment, just like the chloroplast does for the plant cells. So chloroplast is only found in algae and plant cells. It conducts photosynthesis by absorbing sunlight, and it uses water and CO2 to produce the food for the plant. It also contains chlorophyll, which makes the plant green. For the mitochondria, I decided to use a strawberry because just like the mitochondria, strawberries are high in energy. So the mitochondria is known as the powerhouse of the cell. They are gray organelles that act like a digestive system that take in nutrients, breaks down, and creates energy-rich molecules for the cell. So for the peroxisome, these are membrane-bound packets of oxidative enzymes, and it plays a variety of roles, including converting fatty acids to sugar and assisting chloroplasts in photorespiration. It also protects the cell from toxins. I decided to use a blueberry for this because they are similar in structure, and also the blueberry is known for being full of antioxidants to help contribute to good health and wellness. This is the end project of my cell. Thank you.
Why is that so heavy spotted so late? I don't understand. How are people getting hit halfway across the map? I don't understand. Seven heavy shots. Sorry. This guy hates his life. Hit him in the cheeks. SDI shoot? SDI shoot or no? Okay, yeah, that's what I was wondering. Oh my god, he's so heavy to fucking die.
You can see there's the cell wall.
Yeah, fucking missed his shot as well. I don't know if you like lose on your health.
Using lifesavers as the plasmoid matas. Going on the outside. As you can see. I'm using darker green frosting for the plasma membrane.
Try. I don't understand how our whole team died. SC has a red glare. Like legit 755. Steve Force machine.
There's my cell, or my plasma membrane. This right here is my nucleus. This right here, the skin, is the nucleus envelope. The nucleolus is the seed. And then there's the inside of the plum, that is the climactin. This is going to be the rough ER and the smooth ER. I'm going to add some sprinkles to the rough ER to act as ribosomes. So this right here is the rough ER. This right here is the smooth ER. I'm not sure if you can tell, but it's there's little folds in it and stuff. These right here are the ribosomes, the little sprinkles. The ribosomes are free in the cell, so I'm going to add some sprinkles. You can see there's some sprinkles here, here, and here. Pull apart Twizzlers are going to be the cytoskeleton. Pull apart cytoskeleton. The Golgi apparatus. Gummy worms. Mini gummy worms. There's the Golgi apparatus. The central vacuola. So for the chloroplast, I'm going to use food coloring and some green sprinkles. So, it might be kind of hard to see. Right there is the chloroplast with the green frosting and green sprinkles, or green food coloring. The mitochondria. Strawberry. Mitochondria. And the peroxisome, a blueberry. And that is my cell. Thank you.